sharing session. Hopefully, Pak Marvel with the Pacific Scientific, we can do kind of collaboration, with BRIN, do more. This is just first time we can invite your team to do the socialization or even coaching, with our team, because we are consisted of 8 research centers at our research organization. So with 1,000 researchers that actually need this kind of knowledge or technology to accelerate our research. Yeah, so I think thank you very much again, Dr. Nicole, that you are going to share your expertise on NGS with our researchers this afternoon. And yeah, hopefully, Pak Marvel, we can have a meeting in BRIN in Cibinong. Just let me know. I know because in the past 3 weeks we have been very busy regarding the assessment, but I think after this week we can arrange our meeting or discussion to follow up this afternoon meeting, Pak Marvel and Bu Hamidah. Again, everyone, to the committee also, thank you very much for arranging this kind of sharing session, and enjoy. And really, if you have guys any question, just raise your hand or ask the question. And this year again, we are going to run a collaboration platform using this NGS platform, one of which, So, okay, thank you, Pak Marvel, Bu Hamida, and Dr. Nicole. And I give the screen back to Miss Uci. Yeah, so also by saying Bismillah, we open officially this sharing session. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Thank you.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Fathoni. Next is opening remarks from Pacific Scientific to Ms. Hamidah Nur Aulia. Stage is yours.
Thank you for the time, and thank you for our partner BRIN, especially for Bapak Dr. Ahmad Fathoni as a head office in Kepala Pusat Riset Mikrobiologi Terapan. Mungkin saya juga ingin sedikit membagikan terkait profil dari perusahaan Pacific Scientific Indo. Perusahaan kami saat ini sudah berjalan lebih dari 10 tahun. Kami merupakan perusahaan distributor alat laboratorium dan juga reagensia dan juga consumable. Mungkin Bapak Ibu di BRIN ada yang sudah pernah berkenalan gitu dengan Pacific, sedikit juga memberikan historikalnya bahwa kita merupakan distributor terbesar salah satunya di Indonesia dengan beragam brand yang kita sediakan dan kolaborasikan. Salah satunya Twist yang saat ini akan mempresentasikan terkait produk NGS khusus reagensianya untuk bisa disampaikan mungkin di Bapak Ibu yang masih perlu belajar lagi untuk aplikasi di proyek-proyek penelitian di BRIN-nya. Kemudian untuk company kami visinya adalah one stop solution. Jadi untuk seluruh peralatan baik itu reagensia, consumable itu bisa disediakan oleh kami dari Pacific Scientific Indo. Jadi kita melihat personalisasi dulu nih untuk project di BRIN, kira-kira ada penelitiannya apa saja. Jadi bisa kita personalisasi dan bisa kita tawarkan terkait apa yang service yang kita layani. Selain itu kita juga di Pacific Scientific Indo ada quality support, jadi ada maintenance-nya, ada field application-nya juga. Nah, beberapa produk dari kami, ada consumable-nya, dari house brand kami yaitu X-Lab, mungkin Bapak Ibu tahu X-Lab yang mungkin sudah pakai juga, dari microchip, tips, sampai mungkin petri dish, dan lain sebagainya. Untuk peralatan kita banyak sedia dari berbagai brand juga, reagensia, dan juga kita ada service. Kalau misalnya Bapak Ibu di BRIN ada membeli peralatan dan kita butuh tim instalasi dan juga kita ada garansi untuk peralatannya, maintenance peralatan, dan mungkin ada prospek untuk NGS seperti yang sudah disampaikan Bapak Fathoni ya, ada prospek untuk NGS platform. Kita juga ada sedia dari Pacific Scientific Indo yang saat ini baru mulai digaungkan seperti itu. Mungkin sedikit consumable, perkenalan di consumable-nya, kita tadi ada X-Lab, kita sudah terdaftar di e-katalog. Jadi Bapak Ibu juga bisa cari untuk memastikan nih ada nggak di e-katalog, kita ada di X-Lab. Untuk general lab equipment-nya kita juga ada dari IKA, mungkin Bapak Ibu butuh peralatan seperti mikropipet, dan juga ada magnetic stirrer, kita juga ada dari Telstar, itu ada Telstar ada Telstar dan juga ada ALP untuk yang autoclave. Kita juga ada Chemspec untuk spektrofotometer, IKA untuk mikroskop, ada Netek, juga ada Balance untuk analytical balance dan lain sebagainya. Jadi menyesuaikan dengan kebutuhannya. Di life science product di Kota Pasifik juga ada sedia dari Chemometek, ada Azuro Biosystem juga untuk qPCR-nya, untuk bioengineering, kita tadi ada juga Bussiness, untuk kita dari Bioscience, Vitasai, Vivantis, Twist juga yang mungkin nanti akan disampaikan oleh Nicole terkait secara lebih lanjut untuk penerapannya dan apa yang di-provide oleh Twist, terus juga ada Biotech, GSD, Glossar Diagnostik, BRIN, dan juga Science seperti Black Filter. Ada kemudian di Pacific Scientific juga ada provide untuk analytical dan measurement, misalnya untuk water cek water testing, air sampler, dan lain sebagainya. Kita ada water ID, Trailbase, Milwaukee, SI Analytic, dan lain sebagainya. Kemudian ada medical equipment, kita ada dari RAN, gitu, dan juga Cektopa. Ini yang mau saya sedikit lebih bahas lebih banyak ke Bapak Ibu. Ya, jadi di Pacific Scientific saat ini sudah ada provide untuk NGS portofolionya. Jadi untuk produk NGS yang sedia saat ini untuk platform, kita ada dari elemen bioscience, dan untuk reagensia itu ada dari Twist. Untuk workflow-nya mungkin dari molekular bioteknologi, molekular biologinya itu ada dari sample preparation dari Vivantis dan juga PCR Biosystem, kemudian alatnya dari BRIN, ada QC-nya mungkin dari qPCR-nya itu kita dari Azure. Untuk NGS platformnya kita ada elemen bioscience, itu ada Aviti series. Jadi Bapak Ibu kalau misalnya penasaran dan ingin tahu lebih lanjut terkait apa itu elemen bioscience dan bagaimana NGS platformnya, mungkin kita bisa menjadwalkan kembali di pertemuan untuk di BRIN kira-kira mau kapan gitu untuk menjawab lebih lanjut terkait ini. Karena untuk saat ini mungkin berfokus ke produknya ya yang akan dipaparkan oleh Nicole. Untuk elemen bioscience sendiri mungkin sedikit menjelaskan itu merupakan advanced benchtop sequencing dari elemen bioscience itu FPT. Mereka itu dual flow cell dan juga bahkan datanya sudah bisa di standar Q50. Mungkin Bapak Ibu yang tahu NGS dan sequencing saat ini advancement-nya selain dari bisa sequence whole genome, whole exome, di AVT juga bisa multiomics. Nah, Bapak Ibu yang tertarik, jangan lupa untuk isi presensinya untuk kita tawarkan penawaran Twist dan juga NGS workflow portfolio. Kemudian di Pacific Scientific juga kita ada after sale service, dari maintenance-nya juga ada field application support, field engineering service, dan mungkin kita juga ada desain laboratorium kalau misalnya ada buat lab baru, misalnya ada rencana expand laboratorium atau bagaimana, itu kita bisa desainkan terkait laboratoriumnya, mau dari furniture-nya dan lain sebagainya. Flow laboratoriumnya juga bisa kita provide dari Pacific Scientific. Ini sedikit dokumentasi dari pabrik kita, kemudian juga ada showroom terkait laboratorium yang sudah kita instalasi, dari install produknya juga. Mungkin sekian, saya harap Bapak Ibu bisa mengerti dan bisa mengetahui secara lebih lanjut kira-kira di sharing session ini apa saja produk reagensia yang di-provide oleh Twist, dan kalau Bapak Ibu tertarik terkait peralatan, consumable, dan lain sebagainya bisa menghubungi saya di Pacific Scientific Indo, atau mungkin bisa mau berkunjung, bisa kita ada kantornya di Kota Tangerang. Bisa saya kembalikan, terima kasih kepada MC.
Okay, thank you, Ms. Hamidah. Ladies and gentlemen, before we start the agenda, we will have a photo session first. To all participants, please activate your camera and for the committee to take the screenshot. Okay, I will start count from first slide. Ready? 1, 2, 3. Second slide. 1, 2, 3. Third slide. 1, 2, 3. Thank you. And I would like to remind to all participants, please fill the attendance form from the link in the chat box. Now we are going to start our discussion for today, which will be led by our moderator, Marcel Christopher. Before that, I'm going to read his short bio first. Marcel Christopher is graduated with a degree in molecular genetics and biotechnology from Curtin University, Australia. He is experienced in PCR, bacterial and yeast transformations, some exposure to Western blots and RNA extractions. Currently, he works at Pacific Scientific as product specialist. So without any further ado, Mr. Marcel, time is yours.
Thank you. Good afternoon, everyone, and welcome to today's webinar. My name is Marcel Christopher, and I am honored to be your moderator for this session. I thank you all for taking the time to join us. It is a pleasure to introduce our esteemed speaker for today, someone who brings a wealth of expertise and insight to this topic. I would like to welcome Dr. Nicole Lee, a highly accomplished professional in the field of next-generation sequencing and molecular diagnostics. Nicole earned her Bachelor of Science degree in biotechnology from Monash University, one of the top 8 universities in Australia, and has since built an impressive career in genomics and precision medicine. Currently, she serves as the NGS Business Development Manager for Twist Bioscience. She manages countries such as Hong Kong, Taiwan, and a lot of the Southeast Asia region. A little bit about Twist Bioscience. It is a global leader in DNA synthesis and genomics innovation. In her role, she focuses on advancing next-generation sequencing technologies in her region. Before joining Twist Bioscience, Nicole worked with Thermo Fisher Scientific as a clinical application consultant and product manager for, again, the Southeast Asia region, where in this role, she played a very important role in the adoption of next-generation sequencing in precision oncology, enabling the selection of targeted therapies to improve patient outcomes. With her deep expertise and passion for advancing genomic science, Nicole is here today to share her insights and knowledge with us. Without further ado, please give a warm welcome to Dr. Nicole Lee. Nicole, the floor is yours. Thank you.
Thank you, Bob Marvel, for the kind introduction. Let me share my screen. Can you see my screen now? I need to make it to become a presentation mode.
Yes, screen is okay, Nicole. Thank you.
You can see the full screen?
Yes, we can see the full screen.
Okay, good. Firstly, I would like to extend my gratitude to BRIN and also PT Pacific for organizing this webinar. In my role, I collaborate closely with researchers like you across the regions to provide genomic solutions to tailor your specific needs. I'm very excited today to introduce you to Twist Bioscience capability and explore how our technology can empower your research, especially in the areas such as like long-read sequencing, viral and exome enrichment, and also custom panel design. So a little bit of introduction of Twist. Twist is a US company founded in 2013 and IPO in 2018. So Twist actually has 2 manufacturing sites. One is in South San Francisco and the other one is in Portland. So South San Francisco mainly focus on NGS product synthesis, while Portland side mainly focus on the synthetic biology. We do have a commercial office in San Francisco, Tel Aviv, San Diego, Singapore, Japan, Korea, and also China. So who we are? So Twist is a DNA synthetic company. In short, we make DNA for all the researchers. We have 6 different product lines here. On the top one, you see genes, oligo pools, libraries. They are mainly used in synthetic biology research to modify organisms to help addressing challenges in the medicine, agriculture, manufacturing, and also the environment. Just now I went through the video and I heard that BRIN also do enzyme modification. So actually Twist also does help customer to do this as well. So in here, I would like to share more on the NGS panel. So for the oligos that we synthesize, we also can make it become a probe and use it as a capture panel in NGS to enrich the genes or the targets of interest for your next generation sequencing. We also do help our researcher to synthesize antibody. If you are interested, you can contact us as well. So traditionally, oligo was synthesized in the 96 wall plate. So you can see on the left here, the turnaround time for this is actually longer, and also you will use more reagent, which increasing the cost of manufacturing. So now Twist invented actually a silicon platform. It's like a very, very small, like your iPhone size. One time for this oligosynthesis can create more than 1 million oligos per chips. And this in fact is like up 10,000 more scale and throughputs compared to the traditional method. And also Twist does have a proprietary software in the manufacturing so that we will reduce the human error and also error rate in synthesis down to 1 base pair in 2,000 bases. So Twist is using hybrid capture chemistry in NGS. And in hybrid capture, it is very important to look at the capture uniformity. So on the top here, you can see if you receive a panel, inside has a millions or thousands of oligos, they are actually in a different GC percentage. So you can look at the picture here. If the GC percentage is high on that oligos, what happens is that if you use this panel in your capture, you will see a bias in the capture region. Some region will see over capture, which in your result will see over sequence. Some region is under sequence. So Twist actually has a proprietary software to control the synthesis of the oligo frequency to make it very even throughout all the oligos in a different GC percentage. When you receive this panel from Twist in your capture enrichment, so you can see the uniformity of individual regions are pretty much the same. So there is no bias towards certain region. And another thing that I need to highlight to you is every panel that we ship to our customer, we will do a NGS QC on the oligo to make sure that the frequency of the oligos are even and uniform. So I give you some of the example on how we look at the uniform coverage. Let's say, example, we look at an exome panel. Once we sequence and then we normalize the target region. So you can see the profile of individual reads. You will see that if there is a bias towards the capturing process, there will be a higher read step in the GC rich region. However, there is a lagging of coverage in the low GC region. So this is an example that we compare a couple of panels from the commercial. And versus Twist here, you can see Twist is very evenly spread across AT and GC rich region. There is no bias towards high or low. And another one of the parameters that we also comparing is in the hybrid capture is the uniformity on target, 0% coverage target, and target basis as 30x. So all this, when you look at here, everything are a green tick for Twist. So which means that when you use a panel from Twist here, overall, because of a higher uniformity in capturing, so we actually reduce the sequencing cost per sample. How do we do? So when in a run itself, we are measuring on how many samples that you can put in a flow cell. So because of the Twist capture uniformity very high, so each of the sample for Twist actually can reduce the sequencing depth compared to others. So that's indirectly actually reducing a sequencing cost per sample with Twist solution. So Twist continue to innovate our NGS toolset. We launched a viral panel and controls to help address the global pandemic. We also have expanded our solution in targeted methylation, sequencing, and now we're expanding into minimal residual disease direction using liquid biopsy. All the off-the-shelf panels that you see on our website is actually customizable. We can help you to customize every one of the panel into your preferred content. Example over here, Twist also partner with global leaders to develop the exome panel for the nationwide screening program. You can see Broad Institute from US created a clinical research exome using Twist probes and adding more contents to the Twist core exome. And VCGS actually created one of the panel called VCGS exome for a carrier screening in Australia. And for Centogene here in Germany, they created a Centogene exome for rare disease screening. So I also would like to introduce to you Twist Precision DX, a new line of Twist NGS product with a higher quality and robustness for routine clinical use in Europe. So this panel is actually a CE-IVD exome panel. In summary, Twist provide the flexibility to our researchers. So you can come to us on the design. And for Twist here, we do have some off-the-shelf panel like exome, metallome, and viral panel. But most of the researchers come to us is to create their own content. So I would say more than 70% of our customers come to us, they want to do a custom panel with Twist. Twist also provide library prep reagents and also target enrichment reagent as well. And with this, Twist can help in a few areas in your research, like in liquid biopsy, rare disease, oncology, population genetics, infectious disease, and also agri-genomics. I will not focus all in this webinar. I will mainly focus on the infectious disease over here. So infectious disease, always there is a question to a lot of people is why you want to use a respiratory panel instead of a single viral panel. So of course, the advantage here is you can identify core infections. Right now, even though we have a symptoms like SARS-CoV, but however, it could be a mix of between a flu virus and also a COVID. And also you can identify more different viruses quickly compared to a single viral panel testing. And how about a viral panel versus traditional PCR method? So PCR method has a challenge to identify new pathogen. And also PCR, because of the rare of the primer design, you are unable to detect a rare viruses over here. Let me tell you why. So in 2021, when there is a pandemic of SARS-CoV, WHO actually released a note. They compare the PCR and PCOM-based approach versus the capture-based approach. And they realized that actually capture-based approach can tolerate sequence differences from the probe sequence about 10 to 20%. However, MPCOM-based approach is not able to tolerate this. Let me tell you why. So we have tested our panels, our probe itself. Let's say today in the viral, it always evolves. So every single region that you target, they might have a SNPs changes in the virus. If we compare how many our probes itself can tolerate how many different mismatches. So over here, you can see that based on our testing, up to 5 different mismatches, the binding of the probes and the target is still 100%, which means it's still very effective. But let's say you have more SNPs happening in that region, so the binding efficiency will decrease accordingly. So you may not understand what is hybrid capture. So I pull out one of the workflow of the hybrid capture here. What is the difference is that you prepare libraries as normal. Individual sample have been indexed with the barcode. You pull them together, and then now you add our Twist DNA probes into it. So this DNA probe itself is come with a biotinylated end. And when there is a matching error of the DNA and also the probe, then it will bind together. After that, about 16 hours hybridization, or you can choose for a faster one, which is a 4 hours hybridization buffer. After hybridization, so you will use a strata writing beads to pull down the region that actually you want to enrich, and those that do not bind with the probes will be washed away. So at the end, you will receive libraries that actually the targets that you want, and then you can move on to sequencing. But now come into the panel. So what is it, and then how can I improve on that binding efficiency? So on the viral panel, especially because virus always evolve. So let's say, for example, today I have a SARS-CoV viruses. Overall, I pulled out from the database how many strands. Let's say there are total 382 strands of virus. I can create this panel. 100% match is 382 probes. But however, virus always evolve. My probes is only can tolerate 5% if 5 mismatches. But now in our algorithm, design algorithm, we can actually increase random mismatches in the probe. So now you can see from 382 probes now increased to 14,000 probes. And also we also need to cater the way that the evolution of the virus, it could be a continuous mismatch. So now we also can input this into our algorithm and increase more probes possibilities in that case. So and give you an example for a small viral panel like a 29 human respiratory viruses over here. You can see we capture coronaviruses, influenza viruses, paramyxo viruses, and also enteroviruses. With this, actually, you don't need to have a lot of probes. But now you can see example I pull out to you. We use 18,000 probes to capture these 29 viruses. We also adding extra probes actually to target against influenza virus A and B. Why? Because they're more diversity compared to others. They always evolve. And we also have extra probes that actually against rhinoviruses type A, B, C. So total this panel will have 40,000 probes in this panel just for 29 viruses. How about in primers? Primers will not be able to develop so many primers because primers only target on the conserved regions. And there will be no competition in the probes also binding because they won't kneel to each other. So we also add on, make it more sensitive. So all the probes here, we also 4 times increase on the frequency of the probe so that we can capture the very low abundance of the respiratory virus in our panel as well. To add on to this viral panel, we do have a one codec analysis downstreams to help on mapping the viral genome to give you the reports on what are the viruses present in the sample. So you will receive a list of the reports saying that how many reads are belongs to influenza A, how many reads are belongs to the enteroviruses. The next is we do have another panel. We call it as a comprehensive viral research panel. We understand 29 viruses maybe is too small. Some people would like to use a panel that can do a surveillance screening. So this panel itself can cover more than 3,000 viral genome. It includes single-stranded DNA viruses, double-stranded DNA viruses, and also double-stranded RNA and single-stranded RNA viruses. And this panel itself also can capture novel variants of the virus. How can we do it? We also do the strategy that I explained to you. We insert a lot of mismatches into our probe. This panel itself, you look at it, it's only 3,000 virus, but in the content of the panel actually has more than 1 million unique probes. And on top of that, we do support this panel together with one codec analysis. So now you can use this panel to do a virus surveillance screening. And you can see a very nice report to tell you how many percentage of reads map to influenza A, H1N1, papillomavirus, and influenza A viruses also. So you will see this abundance rate count over here on the one codec. And it's quite easy to use on the one codec. Once you sequence, you get the FASTQ file. You just upload the FASTQ file and one codec will actually do the analysis for you. So let me share with you some of the example or how our customer use this comprehensive viral panel in a wastewater surveillance. During pandemic period, in this researcher staying in Houston, they actually collected a few areas, a household area on the wastewater. They want to understand what are the human viral community in this area. So they sequenced all these samples and they realized that using a comprehensive viral panel, they can remove the host because we only focus on capturing the viruses. And on top of that, they can actually enrich 3,000-fold of the virus rates compared to no enrichment. And on top of that, they can identify 450 distinct pathogenic viruses from 28 families. And you can see here, there are a few different areas. There are some areas with abundance, different diversity of the viruses. Some area has a lower. So they can actually see how this abundance of the human viruses is consisting with the regional spread and evolution of the virus. Community with a heavy human activity or staying at the area, they will have a more virus abundance compared to a less crowded region. So this panel also can be used in the wastewater surveillance. And another study is actually from Yunnan province, China. They want to understand how this rodent, which is rats here, actually spreading the virus around the human communities. So because rodent actually is a reservoir for zoonotic viruses, they sometimes including some harmful pathogens such as arena viruses and hantaviruses, yet we don't know about it. So this group of researchers actually interested to understand the wild and urban rodents populations in the Yunnan province area. They collected 124 rodents and taken 681 organs from this rodents from different areas. They use our comprehensive viral panel to actually do enrichment. And they found that there is a high level of human viral diversity in the rodents. You can see here, this blue color, that is actually significantly have a lot of human activity, which is a crowded area. But when you compare at the rubber plantation and other regions that is less community, they actually don't carry a lot virus over here. So this show that the panel show very positive correlation between the mammal viral diversity and the landscape that we studied. And the human activities also may influence the diversity of the virus over here in the rodent population. And the next here is also trees also can help customer to create a completely custom panel. This is more interesting is in Australia, they want to study more about the host pathogen called evolution dynamic. So this Tasmanian devils, this animal, they actually has a disease called devil facial tumor disease, which is very cost deadly. So 100% no cure. So they want to study the group that wild and healthy versus the one that infected, how this animal can transfer the infectious cancer to another. So we help the customer to create the 197,000 probes to design on the SNPs of the genome of the Tasmanian devil and also to look at the genes that cause the changes of the genome after they infect the devil facial tumor disease. So you can see here, during the collection of all these samples, they are be able to identify the regions where the animal carry a lot of the mutation of this disease in the genome. So that is how they see a spread across those positive versus those wild type region. On the comprehensive viral panel itself, we are not only using on the wastewater surveillance, we do have some of the customer will use it for clinical research as well. So in Australia, like Doherty Institute, they actually use this panel to do a routine screening on the samples they receive in the lab. And in the clinical wise, we have a Prince of Wales Hospital and also Widrew. They're also using this comprehensive viral to enrich those virus genome for a further study in clinical use and also diagnosis. In the APAC region, we do have a Korea, KDCA, actually using the comprehensive viral panel for disease control and surveillance studies. And Institute Pasteur du Cambodge in Cambodia, actually also using this comprehensive viral panel. They are using in both on the short read Illumina and also actually also on the ONT platform. In Siraj Hospital, Thailand, they're also using a comprehensive viral panel as well for a research study. So if you want to know more about this comprehensive viral panel, we do have a webinar from George Hawara, from Tohadi Institute, and also from about the University South Health Sydney, Kiwo Kim. He also using this comprehensive viral panel in a clinical patient's diagnosis. So for the twist infectious disease solution, we do have a single panel like SARS-CoV panel. We have a small respiratory viral panel for 29 viruses, and we do have a comprehensive viral panel. So most of the customer also will come to us. They actually want to create more content in the panel, like they want to include virus, bacteria, fungi, parasite. So we are welcome for the customer or researcher reach out to us so we can provide a solution for you. And in the custom panel, we do have 4 different solutions for our customer. The first one is more focused on the minimal research disease, Rapid 500, which is shorter turnaround time for cater on the liquid biopsy monitoring screening. We do have a standard custom DNA panel, RNA panel, and also methylation panel. So for this custom panel itself, on the design-wise, we will have an interactive design with our bioinformatician. So you just need to let us know your genes and the target's information, then our bioinformatician will reach out to you. And there's no limit on the target. You can put as much as 1 million probes over here. And the turnaround time will be very short. So to submit a design request, there are some of the form. Here you can see a DNA form, RNA form, and also methylation form. You can fill up the form and email to me. You say, hi, Nicole, I want to know more about this design form. Can you help me? I also can schedule a call to explain to you how to fill up the form. So once you complete the form, you can send it to me. My email is there. Either you are working on the long or short read panel. Important thing is you need to give me the gene names and also from which species. If it's human, then you need to give me whether it's a H19 or H38. So you send to me, I will send to my bioinformatician team. So they will take a look. If everything is good on the form, he will start the design. So once the design is complete, we will return the design file to you, and then we can have a call to explain to you the design itself, what we cover, what is missed. The next thing here is twist panel is not only support on Illumina, we also do support on other platforms like Element Bio, MGI, and other partners as well, like PacBio also for the short read. And we also able to use the same panel on the PacBio long read and also ONT long read as well. So just share with you on the announcement that we partner with PacBio, and we do have 2 panels, the dark gene region, to cover 389 genes that are very difficult to map in a short read. And also we do have a PGX panel as well on the long read. It's a launch product. If let's say today you want to develop a special small panel like a breast cancer panel only focus on BRCA1/2, we can help you as well. The workflow is pretty similar to the short read. It's just that you will share the DNA in a longer fragment. So with twist, we only focus on 3 factors here. Greater flexibility for our customer, so you can create a small panel to the large panel. You can use it for long read or the short read. And the performance of twist probe is higher and more uniformity, because twist here, I forgot to mention in the beginning, is using a double-stranded probe, DNA probe, and also every single panel, we will do a NGS QC. And lastly here, because of the maximum capture efficiency and uniformity, the sequencing cost per sample is lower compared to other. And twist not just only having an NGS panel, we also do have a control as well. So if you are interested with the respiratory control, or you want to synthesize a new control, you can reach out to Twist. So we do have a list of respiratory viral control, SARS-CoV control to provide you to do an assay validation. And this control is not just for NGS. You can use a control for a real-time PCR assay validation as well. If this is my last slide, and I would like to stop here for Q&A.
Thank you, Nicole, for the presentation. I would like to open to the audience if there's any questions. You can either ask Nicole directly, or if you'd like to use the chat box, that'd be fine as well. Any questions from the audience? If not, I'll start us off with a question from myself. Nicole, can you please, as you've mentioned, that you can create probes for all sorts of viruses, RNA viruses, single-stranded, double-stranded. Can you explain that a bit further, how you can design probes for both types of viruses onto one panel?
To design, so firstly, you will share with us on the virus strands, either the accession number or from the NCBI. So our bioinformatician actually can take all the information from there and start designing the panel for you. So no matter it's a single DNA viruses or RNA viruses, as long as we have the DNA information, we are able to develop that panel for you.
So very customizable, very flexible in the panel design. Yeah. Any questions from the audience? There's one in the check box here. Thank you, Nicole. How to make sure that the kit from Twist is compatible with numerous NGS platform, and what platform has the best compatibility? Thank you, Sugiono, for the question.
Okay, good question. So in Twist, we provide the UDI is a TrueSeq compatible UDI. And if you want to use a Twist workflow, full workflow, is actually the best is on the Illumina. But however, we also use this library, you can do a conversion. Let's say, for example, today you have an MGI platform in the lab. There is a conversion kit, can convert the library into a MGI library. Same goes to PacBio as well. And for Element Bio, we also partner with Element Bio here. Element Bio has launched a new flow cell that is called Flexi flow cell. They can use the Illumina libraries directly. So it shouldn't be a problem with all different NGS platforms. And in fact, our customer also use the TrueSeq libraries to run on ONT as well.
Thank you, Nicole, for the answer. A few more minutes we have for Q&A. We'll just wait if there's any more questions from the audience.
Excuse me, I have a question.
Yeah.
My name is Lara, and I'm just wondering about the detection limit. I mean, if we are talking about a virus, especially in environmental sample, it's really difficult to track. Sometimes we are using NGS, but we are looking also for virus, for example, in wildlife. But sometimes also the NGS cannot really capture the viruses. I'm really curious about like what is the concentration limit for it to be detected in this panel? Could you detect the virus from environmental sample, for example?
Good question. So the detection limit our customer tested, at least you need to have a 25 nanogram of the RNA. The best is a 50 nanogram for both DNA and RNA. So once you have the concentration with this, then we will build the library. So in the building of library, there is an amplification step. So we do amplify the content of inside your sample. So once you have that, and there is some QC step as well, the following. So we do have recommend on after library, after post capture, you will have at least 15 nanogram per microliter. So once you get this, there will be a very good content to put on the sequencing. Yeah.
So you need to know like the concentration of the virus that you have for it to be working in these panels, I guess, like more or less.
Yeah, so for the input, you need some DNA. So we don't count the virus. So when you collect wastewater, so basically you do extraction is using a total nucleic acid. So once you get a total nucleic acid, we just measure on the input DNA that you have.
The total,
So that,
yes,
correct. So that 15 nanogram is inside that extraction.
Okay.
So then after that, we will start building the library.
Okay, and I'm just wondering about the price, like if you want to like make some panels, like for example, I don't know, containing 20 or 25 probes, how expensive it is?
For 20,
per probes, for example, because I know that some people in our institution are trying to detect some bacteria or virus, but how expensive is it for the panels? Just curious.
So we have a different tier pricing. So the lowest one will be minimum about 500 probes.
500. Oh, okay.
Yes,
500 probes.
correct. So you can put more until like not just 50, you can put all the way to 500.
Okay, and how is the price? I mean,
the price
for 500 probes.
I would say you share with me the content one time, then I will share with you how the price look like, because there are some of the way that we can help to actually optimizing on the price at your screening.
Okay. So also you could mix like like between the bacteria and virus, and also for the fungi, it could be mixed, right?
Yes, could be mixed. Correct.
Okay. Yeah. All is clear. Thanks a lot.
Thank you, Razara, for the question. Very good question. Any more questions from the audience? Another one here from Pak Sugiono. Terima kasih buat pertanyaan Pak. According to your experience, any non-targeted sequence that are amplified using the kit, for example, the host?
Okay, yes. So sometimes you will be able to capture the host. So because our panel itself has more than 1 million probe inside. So if there is a similarity sequence for host and the probe, you will be able to capture. But however, at the same time, we actually focus on enriching the virus. So we do have non-targeted sequences in your output. But at the same time, we also see actually the enrich of the virus. So the host itself is still very small percentage, maybe 20% like that throughout your capture. Have this answered your questions?
Yes, thank you. So what you said, it's 20% the virus? Oh, that's quite high actually. Compared to our experience using other kits from Illumina.
You mean 20% for the host or?
From for the virus?
Okay. So viruses actually.
For the virus itself? Yep.
Correct. We can capture about 20% of it.
Okay. That's quite high.
Thank you, Pak Sugiono, untuk pertanyaannya. Nah, ada lagi dari peserta yang mungkin punya pertanyaan? We'll give a few more minutes for the audience to submit some more questions.
Can I ask another question?
Yes, of course, sir. Please go ahead.
For the codex, is it compatible to analyze from various NGS platform as well?
Yes, you can analyze from various NGS platform as long as you can generate the FASTQ file.
Yeah, okay. With the FASTQ. Okay.
Yeah, correct.
Okay. Right.
Thank you, sir, for the question. We are ahead of schedule. So we still have a few more minutes for the questions, but If there are no more questions, maybe I could hand it off to the MC. We'll say a few more, 5 more minutes for the participants to submit questions.
I think I have a little bit more questions. I mean,
Yes,
for the
please.
probes, for the target, is there any limits of the size that we could use? For example, like the base pair, the maximum, the limit, the size limit of the target sequences.
Good question. So our probe size, the length is 120 base pair itself. So for the library inserts, we can capture as long as you have more than 120 base pair.
More than 120.
Correct. So let's say you have a sample DNA, then you will fragment the DNA into a smaller size. You will share the DNA around 300 base pair like that. 250 to 300 base pair will be the good one.
So also it depends on the sequencing platform then.
Correct. So short read
Okay.
normally we'll recommend around 200 base pair to 300 base pair.
Okay. Thank you.
Question?
Yes, there's a question from the audience. I heard somebody say something.
Is it okay if I talk a little bit more?
Yeah.
So I'm also working in the sequencing facility in BRIN. So we have sequencing platforms such as ONC, Illumina, and also MGI. And the problem is, I'm not really sure because this is also new technology for us, these panels. Could you provide some like flyer or something, the data that we could use, I mean, for each of the sequencing platform like Illumina, ONC, or MGI. So maybe also not only the Research Center for Applied Microbiology, but we could also expand it to the other research center, for example, because BRIN have different sequencing platform like Illumina, MGI, and also ONC, and I think we could also use this platform also in combination with this panels, I guess.
Yeah, definitely.
Maybe if you could,
I can, sure. I can share more information on that part, how our panel can use in different platform. Maybe it will be good to have a separate call so that I can explain on the workflow to you.
That would be great, yeah.
Yeah, maybe Marvel, can you help to schedule another meeting with the customer?
Yes.
So I can follow up with that. So for myself, I support the researcher across the region also using different platforms. It's like for MGI, a lot of customers will build the library prep according to our protocol. Then they just do a commercial kit and they can sequence on MGI without any issues. ONT, I would say, because ONT is still new, the key thing is on the demarxing. So there is a online tool called Doroto, recommended by ONT. So they can actually demarx twist through SIG adapter. So once you demarx, then you do alignment. After that, you actually can submit to the one cortex also when you successfully generated the FASTQ file.
Question, another one, Nicole. After this webinar sessions, will there be any workshop hands-on about this technology?
Sure, we can organize a hands-on workshop whenever you have available slots. So we can arrange some reagent and show you how to do the hybrid capture.
Yeah, I think that would sound lovely.
So when will be a good time for us to come down to show you?
Actually, can I speak? Perhaps we can schedule it after this study session with Mr. Ahmad Fathoni or all the people that have list in the presidency. We have registered and we can share and also request for the further schedule for the meeting with BRIN or something with you. Thank you.
Yeah, I think that's good idea. Yeah. So if you can follow up this webinar with hands-on workshop, so in more detail, I think yeah that will be great. So because in BRIN we just created new unit actually under Deputy for Infrastructure, focus on capacity building, yeah, including a workshop training. So please Pak, maybe we can have another meeting ya Pak Marvel and Bu Hamidah ya. We can arrange make a deal when we can arrange the workshop maybe next month or in March ya. It depends on you Pak Marvel and Dr. Nicole ya when you're available. We are actually quite flexible and then what we can support is like we collaborate with Deputy for Infrastructure and then facilitate what we need actually like a room or equipment, something like that. Yeah, but I think we need to have agree another meeting in more detail Pak Marvel. If you can visit BRIN maybe next week after a long holiday because next week we have long holiday, really long holiday after Lunar New Year, and we have we can discuss in my office to discuss in more detail rundown and etc. But okay, let's say we can propose to Deputy for Infrastructure in March. We are going to run like a hands-on workshop on this kind of technology, and then I can announce to the Deputy first if we are going to run this. So yeah, should be okay. Yeah, I agree with that plan.
Yes.
Yeah.
We will follow up on that after
Okay.
long holiday and we can discuss further. I think that sounds like a very good idea, and
Yeah.
we should arrange as soon as possible while we have the ball rolling. But
Yeah, so Pak Marvel or Dr. Nicole, so is it possible for you guys to like send like a letter to us? Yeah, it can be to me for our plan to do or to run on hands-on workshop. So I can use that letter from you to proceed further in BRIN. Yeah.
Okay.
Okay.
Yeah,
So and one more thing here we have Dr. Zahra is she has some question, but she's one of our key person actually in sequencing program in this year, and hopefully Dr. Zahra later on can provide you in more detail about our platform in BRIN. So from that we can discuss more, not only in the virus virus field, but beyond that because we have various samples from animal, plant, human microbes, and not only virus but all microbes that we need to characterize using this kind of technology. Yeah, hopefully we can broaden the area, the spectrum, and we once you later on see our facility and you can give an input what kind of research that we can maximize using your technology at Twist Bioscience and or PT Pacific Scientific. Yeah, I think that two things that we can collaborate and follow up from this webinar. Thank you.
Yeah, thank you, Pak Ahmad. And it sounds good to me. It's very good to have an idea to follow up with a workshop. I also will have a separate call with Dr. Zahra to understand the equipment in the lab
Yeah,
that
yeah.
we can prepare the workshop better.
Yes, please. Yeah. Okay, cool. So, yeah, let's do it.
Thank you very
Okay,
much.
thank
right.
you
Thank you
Pak
very
Ahmad.
much.
Ntar kita kontak-kontakan lagi Pak
Boleh,
for
boleh.
follow up. I think it is a great idea and very excited to get that going. Yes, see how we can collaborate further and like as Pak Ahmad mentioned, there's a lot of samples, a lot of different sample types, and I think Twist has the flexibility to provide with many needs of brain. Yes. if there are no more questions, maybe I can hand it off back to the MC for closing remarks. Ibu Arini.
Okay, thank you, Mr. Marvel, for leading a very interactive discussion. And thank you, Ms. Nicole, for a very interesting presentation. And I would like to remind once again to all participants, please fill the attendance form from the link in the chat box. Ladies and gentlemen, we have finally come to the end part of the agenda. Before I close this session, I would like to ask Head of Research Center for Applied Microbiology, BRIN, to give us some short closing remarks to Dr. Ahmad Fathoni. Time is yours.
Thank you, Ms. Uci. Again, thank you very much, Dr. Nicole, for your experience, and also Pak Marvel, Bu Hamidah, for today, arranging this kind of really valuable webinar, and all participants, and emphasize two things after this. This is not just sharing session and then stop and gone, but we want to follow up this kind of meeting to speed up our research using next generation sequencing. The first one, we are going to have a meeting in more detail with maybe later on with Dr. Zara and Dr. Sugiono. And then we can introduce our platform in BRIN, and then you can give input how we can optimize our platform here using your technology at Twist Bioscience. And the second one, hopefully we can run a hands-on workshop maybe in March, but we can set up from the beginning right now after this meeting how we can step by step to do hands-on workshop. And maybe we will create team to conduct the workshop with your team, Pak Marvel and Dr. Nicole, just to coordinate until we run the workshop later on. I think these two things very fruitful meeting for all of us. And thank you everyone for attending this webinar, and see you in the next occasion. So I close this session. Thank you everyone. Assalamualaikum warahmatullahi wabarakatuh. So please, Ms. Uci, give the screen back to you.
Thank you, Dr. Fathoni. Ladies and gentlemen, this is the end of our agenda for today. I'm Anelzi Arini. On behalf of the committee, I would like to say thank you very much to our speaker, moderator, and all participants. See you in the next event. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Nicole, everyone. Bye-bye. Thank you, Pak Marvel, Pak nanti kita follow up lagi aja. Kontak Pak Sugi atau nanti dengan Bu Yara kita ada meeting.
Iya Pak, saya apakah boleh izin minta kontak Bapak? Saya belum ada Pak.
Oke, boleh.
Masih ada direct message di chat box juga.
Udah, Oke, nanti apa namanya, Mas Ugi udah ada ya, Pak Sugiono sudah banyak kontak ya. Nanti saya kontak itu. Bentar, ini langsung kasih itu aja number saya ya. Oke.
Terima kasih Pak Ahmad atas waktu. Terima kasih
thank you
atas
Pak Marvel
kesempatannya untuk menjadi moderator juga.
Bu Hamidah, teman-teman semuanya
banyak,
Mbak Lia, terus Mbak Uci. Oke,
terima kasih semua komite. Terima kasih kepada BRIN atas kerjasamanya.
Oke.
Terima kasih banyak.